Good afternoon. I'm Michael Lai. I'm a virologist, but I used to work on hepatitis viruses. And for me, influenza virus is a new virus for me, so I come here to learn, not to lecture. I'm sorry that I'm losing my voice, so I have to speak softly. So in case you could not hear me, please remind me, and so that I can speak closer to the microphone. What I'm going to share with you today is a study of the host factors for influenza virus replication. Influenza virus, everybody knows, is an RNA virus. It has 8 RNA segments. Each RNA segment code for 1 or 2 proteins. So 8 RNA segments calling for 12 proteins. Some of the proteins are structured proteins, and some of them, all these are structured proteins. And the non-structured proteins are for RNA transcription, RNA replication, and non-nucleocapsid protein and the polymerase proteins are responsible for RNA synthesis. So this is for viral RNA synthesis. The structured proteins are responsible for synthesis, production of virus particles. And there are some additional proteins, non-structured proteins, mediate suppression of host cell antiviral responses. The virus infects the cells through the receptors binding to salicylic acid as a receptor. Then the virus particles is taken up by the cells by endocytosis. Then virus particles, so the virus gets into the cells through the endosome. Then the endosome membranes and the virus membranes form fusions and then release the virus particles, virus internal components, viral RNA and viral proteins. This viral RNA then goes into nucleus where RNA transcription and replication take place. Then the viral RNA will go into the cytoplasm where proteins are made. Then proteins, some go back to the nucleus, some stay in the cytoplasm where the virus particles are made, and then virus particles are released from the cells. So influenza virus is very unique in the sense that it replicates in the nucleus. So this is very rare for RNA viruses. So how does the virus get into the cells through endosome? How does the virus get out of the endosomes to go into nucleus? That is still not clear. And it's probably because the virus genome is so small. So most likely the virus genome does not call for enough genetic information. So the virus replication probably needs cellular factors to complete this process. So the study of cellular factors becomes a very interesting subject because the antiviral components, antiviral drugs, the currently available antiviral drugs target the viral proteins. But the viral proteins mutate very quickly. So alternative approach is to target the cellular factors which are essential for virus replication. So it would be very good to study to understand host factors. There are many ways to study host factors, but now the currently very popular way, one of the very popular ways is to do RNA library screening. Use RNA library screening. Use shRNA, the inhibitory RNAs, and then make the RNA library, transfect the library into the cells. Usually the traditional way is use array RNA clones. But that method is very expensive and very time-consuming. So we modified the methods, use a pool shRNA library, and transfer into cells. Then using the cell population, pooled populations infected with influenza virus. If the cells express the RNAi, inhibitor RNAi, essential for virus replication, then these cells would not be infected by the virus, so the cells will survive. So the cells will now contain the genes that are essential for virus replications. So now you can isolate the genes, sequence the genes, and you can immediately know what genes they are. So by using these methods, from 20,000 cellular genes, in the first screening, we identified around 100 genes that are essential for RNA replications. So among these RNA genes, you can see just an example. This is ATP genes. And this is the infected cell, the wild-type cells infected with influenza virus. Now you can see they all express the nucleoprotein. But if this gene is essential for virus replication, so they cannot replicate, the virus cannot replicate in the cells. So we know this gene is a very essential gene. So by using these methods, we identify about 100 genes, and these 100 genes are all important for virus replication. And the one I'm going to share with you today is each gene. What is each gene? Each actually is this way, about 100 genes. There are many different types of genes. There are membrane proteins, the membrane protein, and all kinds of genes. But what I'm going to share with you today is the ubiquitin-dependent genes. Why ubiquitin genes? Because ubiquitin is very important for the biological functions of the cells. Ubiquitination system involves 3 different enzymes. The ubiquitin activating enzyme, G2 ubiquitin conjugating enzyme, and the ubiquitin ligases. Now I put the ubiquitin onto the proteins. There are 2 types of ubiquitin chains. One is the lysine 48 chain, and this now will be degraded by proteasome. And the second type of ubiquitin is lysine 63 chain, and this becomes a important signaling molecules. So ubiquitin is very important for maintaining the metabolism of the proteins. And we found that one ubiquitin ligase, that each ubiquitin ligase, each ubiquitin ligase is important for many biological functions. For instance, regulation, receptor trafficking, immune responses, regulate cell death, differentiation, transcription, endocytosis. So each ubiquitin ligase is very important. Is this important for virus replication? Yes, because we selected it by its resistance to virus replications. So it's important for virus replication, but how? So now you can see that this shows that this is each expression, the shRNA expressing each RNAi. Now you can see the nuclear virus cannot replicate in the cells, cannot infect the cells. So the MP protein is inhibited. And so we know that each is important. Each ubiquitin ligase is important for virus replication. But how does it do it? And this is, if you look at the virus infection, virus entry to the cells, you can see this is, look at panel C. 10 minutes after virus infections, most of the viral proteins, about 10% of viral proteins is in the nucleus. The rest are in the cytoplasm. But after 60 minutes after virus infections, now 40% of the viral proteins are in the cytoplasm, in the nucleus, I'm sorry. So 40% in the nucleus. So you can see the viral proteins, virus gets into the cells, Now the virus proteins move from the cytoplasm into the nucleus. But if we express each RNA inhibits RNA in each RNA, now it doesn't move into the nucleus. It stays in the cytoplasm. And we know that it stays in the cytoplasm, not freeze in the cytoplasm, but stay in the endosome, So it doesn't get out of the endosome. That's why now it stays in the cytoplasm. Now it doesn't go into nucleus. So that tells us that each ligase, ubiquitin ligase, is important for the virus RNA to get out the endosome to move into the nucleus. So this is how we prove that there are some other methods, some other evidence to support this hypothesis. That is that each ubiquitin ligase is important for the virus endocytosis, important for the virus RNA to get out of the endosome and to move into the nucleus. So how does it do it? Each ubiquitin ligase, actually we found that it binds to the matrix protein, M1 protein. I need to show you the methods, but I want to show you the results. The M1, it binds to the M1 protein. So it binds to the matrix protein, and that makes sense. Matrix is on the outside of the virus particle. And how does it do it? You can see that each protein actually interacts with M1 protein. And ubiquitin is M1 protein. This is ubiquitination. If we express each RNAi, now the ubiquitination is inhibited. So this shows that each now we can say that each interacts with M matrix protein, the ubiquitinase M1 protein, and then that allows the virus particles to be degraded and then to get out of the endosome. That is how we do it. So we know that it is involved in virus uncoating. Uncoating, that means that the virus RNA proteins gets out of the envelope, and that is called uncoating process. So this is uncoating during the virus entry. So how does it do it? The M1 proteins gets into the endosome together with virus particle. Then next is activated by phosphorylation of each phosphorylation. Each now is activated. Each now is ubiquitinate the matrix protein that allows the virus particle to get out of the endosome and then moves into the nucleus. So this is an essential process of the virus entry. That is how each ubiquitin ligase participates in the virus replication. Okay. So this is the first ancillary factors we identified. There is each ubiquitin ligase. So the next step now, the viral RNA now gets out of the endosomes, goes into the cytoplasm. Now it moves into the nucleus. Now where the RNA replication and the transcription works. So you can see now the viral particles gets into the nucleus. That's all right. So it cannot. So once you get to the endosome, now it moves into nucleus. Nucleus where RNA transcription and replication takes place. RNA transcription and replication actually is different in influenza virus, and transcription and replication means the same thing in other RNA viruses. So in influenza virus, it's different. Because the transcription means making messenger RNAs, because influenza virus use very strange mechanism to make the messenger RNA, and use the different mechanism for making replication. How does it do it? The influenza virus uses cellular messenger RNA. Messenger RNA. do we have addition pointer? Okay, the influenza virus use cellular messenger, free messenger RNA. Use free messenger RNA as a primer, and then so the free messenger RNA is. Oh, thank you. Free messenger RNA is a primer, then it remove about 13 nucleotides to serve as a primer for transcription, for messenger RNA synthesis. This is transcription process. And so this is called capsid snatching mechanism. Okay. And so you can see now transcription for replication of the messenger viral RNA is still, the mechanism is still not quite clear. Now we found that one protein actually is important for the process. This protein is called RP1B. There's a ribosomal RNA processing, one homolog B. This is a nucleolar protein involved in ribosome biogenesis. But so you can see if we express the RNAi of RP1B, this nucleolar protein, now virus cannot replicate in the cytoplasm, cannot replicate. And we found that it's important for RDRP, the RNA-dependent RNA polymerase activity, RNA-dependent RNA polymerase activity. Okay. And then it's important for using primary extension study. We can see that it's important for messenger RNA synthesis. This is a wild-type messenger RNA. You will express the shRNA of this protein, of these genes. Now the RNA, messenger RNA, it becomes much weaker. So that means the messenger RNA transcription needs these cellular factors. And because I don't have enough time, I don't go into details. We can show that by inhibiting the virus RNA replication using protein synthesis inhibitor cyclohexamide. Cyclohexamide will inhibit the RNA replication and so allow us to study the transcription only. So using this method, we inhibit the cyclohexamide, inhibit translation. Now the replication cannot take place. So the replication cannot take place. So we can study the messenger RNA. As I told you, the messenger RNA transcription is different from replication. So now we can just look at the messenger RNA. The messenger RNA, you can see. You can see the messenger RNA. This is this transcription is dead. But anyway, that transcription is the what type messenger RNA. Messenger RNA, wild type RNA here. You can see, and there's a RNA I treated the messenger RNA. So that shows that messenger RNA is inhibited once RP1B is not knocked down. So this is RP1B is necessary for transcription. And you can see this is, again, the wild-type RNA now is darker than the mutant genes. So this mutant gene is important for transcription. And for viral replication, then there's no difference. So this shows that this one is specifically required for transcription, but probably not for replication. And how does it do it? We can show that in the right panel, you can see there are 3 proteins that interact with RP1B. There is a PV2, PV1, and MP protein. So the polymerase proteins and nucleocapsid proteins interact with this protein. And this protein inhibits, by binding this protein, by binding into the proteins, the RNA polymerase activities is inhibited, so the transcription cannot go on. And this is a detailed mechanism. I don't have enough time to show you that. This data says that this RP1B binds to the cap of the messenger RNA. The cap sequence of messenger RNA inhibits the transcription by binding to the capped messenger RNA. That's why the transcription cannot go on. So you can see. This is the capped messenger RNA here. And this is uncapped messenger RNA here. So the capped messenger RNA, protein will bind to the capped messenger RNA, and then allows the capped messenger RNA to serve as a primer for transcription. So that's how it works. And this again, they show that if we inhibit the expression of these genes, RP1 gene, then the amount of capped messenger RNA binds to the polymerase is reduced. Therefore, the transcription is inhibited. So that is how this protein works. So to summarize what I shared with you today, I think that we have identified, my laboratory in the last couple of years also have identified at least 4 genes that are important, cell factors that are important for virus replication. There is a ubiquitin ligase each, and this is involved in the virus uncoating by binding to the matrix protein and by ubiquitinate the matrix protein M1. The second enzyme is, I didn't report to you, there is a D-ubiquitinase USP11 that inhibits RNA replication by ubiquitin MP protein, D-ubiquitinase MP nucleocapsid protein. So this is important for RNA replication. This shows that the ubiquitination of the MP proteins is important for RNA transcription. So again, the third one is the transcription factors, DR1, that can facilitate RNA replication and suppresses the innate immune molecules by binding to RDRP and binding to the cellular RNA polymerase. Now, the fourth protein is the RNA binding factor. The last protein I show you, the RP1B. This inhibits RNA transcription, involved in RNA transcription. So you can see there is a lot of cellular factors that are necessary for virus replication, involved in different steps of virus replication. So by using cellular factors, then the virus RNA can replicate, virus protein can be translated, virus can be uncoated. So all these processes require cellular factors, and some of these cellular factors will be ideal targets for antiviral drug development in the future. Okay, so thank you.